And we are back with our fifth presenter. Our fifth presenter is Jeffrey Sanders. Jeffrey is with Schrodinger. Jeffrey is the scientific lead for the Consumer Packaged Goods Division and has been with the company for 8 years. He received his Bachelor of Science in Applied Physics from Worcester Polytechnic Institute and his PhD in Biophysics and Molecular Pharmacology from Thomas Jefferson Medical College. Jeffrey's going to talk about exploring the structure-function relationships of protein-based surfactants for food emulsions and foams.
Thanks, Ryan, and thanks everybody for attending. So I know this talk might be slightly different than the previous ones, and hopefully, it's interesting to at least a few people who are understanding how, molecular-based simulation can be applied to not just what I'm going to talk about, which are protein-based emulsions, but also a lot of other different types of, oils, condensed phase systems. We can do a lot. And so just to, if you're unfamiliar with Schrodinger, we've been around for about 30 years now, focused a lot on life science and materials discovery, so new material discovery. And, our purpose here really is to improve human health and the quality of life by transforming the way materials, therapeutics, and, new chemistries are designed and discovered. And how we do this is through simulation, and many of you maybe are familiar with multi-physics simulation or FEA simulations. We're on the exact opposite end of the scale. So we're talking about things on the molecular scale. I'll give you a sense of size and length scales, but we're using physics-based simulation much like other computational methods are. And we do this through a variety of applications, some for drug design, some for therapeutic design, but also primarily material science. And so if you've ever interacted with protein crystal structures or small molecule crystal structures, you ever had to make a graphic or opened a scientific journal and seen a bunch of really nice molecular structures, they were probably made with Pymol. And so we are the commercial maintainer of Pymol. It's freely available for academics still. And we tie all of this together either through a core modeling suite or an informatics platform if you're trying to democratize simulation out to a larger organization. And I know that all sounds, very, physics-based, okay, but what are you actually doing? And what are you simulating? So when we think about, the challenges with multi-scale modeling, right, and this is a, cross science and engineering problem, is really marrying the scales between very low level simulations, maybe high level of theory, up to something that is a whole part, a whole vehicle, or a whole product at the end of the day. And that could be almost anything, really. And so where we, exist and where our tools really focus are on the, the electronic and the atomistic time and length scales. So in those cases for electronics, we're usually looking at a single molecule or a periodic structure that's crystalline, and we're using density functional theory. We're using plane wave periodic DFT to describe the electronic properties, and you get a lot of interesting things out of there, UV vis, IR, and a lot of other relevant properties like redox potentials in this case, or bond association energies, especially for lipids, which are super important. And then when you move up to classical based simulation, we talk to atomistic, we're thinking of molecular mechanics, MD. We're really using physics-based simulation to describe using Newton's laws of motion how atoms behave in a liquid phase and a solid phase, and that can interfacial systems, any sort of combination where we can describe the chemistry very well. We can model it. And then ultimately, you want to take that information and put it into something that's much larger, like a mesoscale simulation, whether using Monte Carlo methods or coarse grain, molecular dynamics simulations. And, if you simulate big enough, the idea is that you could take the output of that and use that as something like a representative volume element in a continuum mechanics simulation. So, while this is great, this is still a very difficult problem to think about, just overall trying to use pieces of information from each time and length scale and add them to the next higher one. So ultimately, electronic properties could one day use to be feed into a continuum model that could perfectly describe the electronics all the way up to a whole part or a whole product. But we're, as a community and as a company, we're focusing very hard on trying to make this, something that's approachable and not just something hand wavy that, someone might talk about and then it only works in one particular case. And I like to orient when I'm talking about smaller length and time scales, since this is much smaller than people are maybe are used to dealing with, especially on the manufacturing side. When we think about what can we simulate, what can we look at at the nanometer scale, or maybe the 10 nanometer scale, we're really talking about looking at surfactant condensed phase systems. We're looking at maybe micelles, bilayers, aggregations, whether it's small molecule, large molecule, polymer, protein. And really what we're able to simulate are things up to like actual micelles that you can validate using something like dynamic light scattering, or some other experimental method for critical micelle concentration. There's a whole host of properties I could go on for an hour about. But the idea is that we're really looking at either the interfacial properties of emulsions or foams, or we're looking at very small, maybe nano emulsions. And that's really where we shine and the tools work very well. And just again, to understand the differences here, since I think it is important, since all this is based on chemistry. And something that I think we don't emphasize enough and always comes up in questions after these talks is, what are the parameters that you use to fill your simulation? How do you set it up? We don't use experimental information per se. We're not using a bunch of ODEs. We're not inputting kinetic rates. We're really just using quantum mechanical properties with condensed phase experiments for van der Waals estimations of atoms, and that's all we're doing. So we're letting the chemistry and the physics completely describe the temporal evolution and the electronic properties if we're talking at the QM scale. So there's not a lot of input as long as you can describe the chemistry and what's in your formulation, what's in your oil, what's in your interfacial system, you can most likely simulate it. And depending on how long it takes for an interesting property to evolve, maybe self-assembly, or you might have transport properties. Those are usually something you can either do a simulation, or you can use different techniques to answer those questions. And so, since I'm going to be talking mainly about molecular dynamics simulations, the one thing that's very important is a force field, right? So understanding the chemistry, as I mentioned from the QM Lembel, force fields are a combination of QM calculations to describe partial charges and bonds and all of that fun stuff. But really what we're doing at the end of the day is we're just solving Newton's second law. And we're describing the temporal evolution of all of the atoms in the system and how they behave under certain environmental conditions, whether it's changes in temperature, pressure, or changes in pH of the system. So, laying the foundation for how we would use the simulation, it's in the past 10 years with the advances in computational power. So GPU computing is very popular besides just Bitcoin, mining for everything else. We leverage those to run physics-based simulations at speeds that we would never have been able to do 10, 15 years ago. And so what that means is that we can start looking at properties like self-assembly for something like an emulsion foam. And this is just a simple example illustrating right at the macroscopic scale. Everyone's seen what whipped cream looks like. If you go down to the microscopic scale and you start to look at the emulsion properties, right? So, how do the micelles form, or if there's an air gap because it's a foam, what does it look like on the micron scale? And then what does it look like at the molecular scale? And so, thinking about, we can't simulate all of this, but we can start feeding in molecular pieces using physics-based simulation, and that can help understand the microscopic properties at the end of the day. And since this is just focusing on a protein surfactant, we can also simulate traditional surfactants. We thought this is interesting because this is something that, as food and beverage, cosmetics, and all these industries focus more and more on natural ingredients, understanding where it's come, proteins are becoming more and more a health, choice, or something, for an alternative meat, or something that you can add to your product that is, naturally made, quote unquote. And so there's a well-studied fungal protein. They're called hydrophobins. They're made in filamentous fungi. They are highly amphiphilic, so they have very good surfactant-like properties. They're excellent candidates as emulsifiers, anti-wetting, surface coatings, and also for tons of biomedical applications. So understanding how these proteins self-assemble, how they form interfaces between air or hydrophobic and hydrophilic solvents is really important. And so if we can simulate these and start to understand the mechanisms, that can give us some really useful information if we want to try and develop more complex formulations, or we want to add them to an existing product. And so, the assembly or the stoichiometry of how these work is you have a single molecule, and at an air-water interface, the way that the protein folds is that it has a very hydrophobic patch that will orient itself along either a hydrophobic solvent interface or air, and the hydrophilic just stays in solvent. If it is in a full solvent phase, not in a foam, it will display different properties depending on the solvent conditions and environmental conditions. You can have just a single protein molecule form dimers, tetramers. It can form a polymer, either a nanotube or a fiber, depending on the solvent conditions. And this has, been studied academically for years, and is illustrated by the fact that if you crystallize the protein under different solvent conditions, it will form different asymmetric units of different space groups. And the most well understood one, which is HFB2, hydrophobin 2, has a really nice asymmetric unit tetramer in the crystal structure for one solvent condition. But if you swap that out later, you can still get an octameric assembly that looks more like a polymer nanotube. And so really controlling solvent conditions is going to be very important for how these things assemble, but also when you start adding in either co-emulsifiers, or if you need to add in other solvents, preservatives, what does that do? How does that change the stoichiometry? And how does it drive these proteins to form one type of assembly versus another? And so one tool that we use, and this is for applications in fermentation, where we're trying to understand how a protein might behave when scaling up, when processing at large scale. So does the protein aggregate, and how do we minimize that? So we can use structure-based tools, and this is physics-based because we're looking really at the electrostatic surface and the partial charges along the surface of a protein to understand what portions of the protein are charged. And in certain cases, as I mentioned, fermentation and others, we're trying to understand how to control that, because maybe we care about aggregation. In this case, for hydrophobins, we want controlled aggregation in a very specific manner. Or in cases where you need soluble protein for some application, you don't want it to aggregate. And so Schrodinger published a paper, I think 2018, a few years ago, in a collaboration with BMS, where we looked at, if we make changes to protein sequences to make them more or less hydrophobic, how does that control overall solubility after you make it in a bioreactor? And so this tool was able to properly predict, based upon the aggregation score, or ag score as we call it, how well something will aggregate, or where, what part of the protein surface is actually driving aggregation. So we can apply this to hydrophobins. So this is just using, one hydrophobin structure, illustrating that, the scores correlate very well with where the patches are. So that little red patch on the bottom is actually the part of the protein that orients itself onto the air interface, or if there's a hydrophobic solvent. So that's really nice. Outside of crystal structures, this is, pretty much well understood, but confirming that the technique actually works appropriately. And we can further break down, these scores to look at on a per sequence basis. What's the hydrophobic contribution? What patches of the protein are contributing more, or are going to more likely orient along that interface? And why this is important is you may not want to engineer these proteins, although people have gone to some lengths to add other structural elements to them. You might want to select which protein in particular you want to use. So maybe you're using a different class, you're picking it from a different organism, and you want to understand, what's the relative size of the molecule. Maybe you have another lipid that you want to interact, and you want to know where is it going to interact. Is it going to disrupt fibril formation, polymer formation, or aggregation and self-assembly along an interface, or solvents, or an emulsion at the end of the day. So what this does is allows us to break down on a sequence specific manner what the ag scores are, what the hydrophobic contributions are, and that allows us to make selections. So there's maybe 15, I think now, crystal structures known for different hydrophobins, mutants, different organisms, and being able to understand, identify which ones have high aggregation scores might be a better selection candidate for a particular type of application. So, using a simple screening technique like AgScore, something that takes about 2 minutes to run, we can get some very useful information about these proteins and maybe use this as a course selection guide or criteria. But if we really want to understand, how does aggregation play out in different conditions, we really have to start looking at, simulating these things in solvent. So we can take the simplest one. We can look at, hydrophobin 2. We can look at a tetramer. We know what the crystal structure looks like. We can pop it in water. We can run the simulation for 200 nanoseconds. We could run it for longer. This is just, the shortest amount of time to get some interesting behavior. What we see is that if we start with monodispersed protein in water, it very quickly aggregates. It does form an assembly that's slightly different from the crystal structure, which makes sense because the crystal structure solution was, I don't remember the exact details, but it was not water. There's usually a lot more in there. Usually there's PEG, there's other excipients or co-solvents. And, it actually starts to form a prefibril. So even though we're only explicitly simulating 4 molecules, we're actually, can look at in space, does it align? And it starts to align in somewhat of a protofibril manner, which is really interesting because, that's something that's been suggested by the scheme of how these things self-assemble, but never observed explicitly, at least not outside of one single small angle X-ray scattering paper. We can further take that and we can look at an interface. So does it assemble the same way that it would in water? We don't expect so, because now those hydrophobic patches on the bottom don't need to bury themselves against each other or, try to reduce the solvent exposed surface area. Here we can actually see that if we put something up against like a very hydrophobic solvent against a water interface, that they will align as we'd expect, but they're going to align very differently. They're more spread out. It's more like a linear polymer in that case, even though they're not covalently attached. And so that's, really nice, because while these are simple systems, and while, it's somewhat expected based upon aggregation scores and what's been seen experimentally, this allows you to start expanding, maybe swapping out solvents, maybe adding co-emulsifiers, changing the temperature, heating things up. These are all done at, room temp, these simulations, but you could easily heat them up, cool them down. You could add an air gap in there, a vacuum gap, so that you can look and see how does this assembly change on an air interface versus a hydrophobic, hydrophilic solvent interface. And you can start to add more and more molecules. So, since we can run these simulations for a long period of time, or we can run larger and larger systems, we can keep adding more molecules. We can look at, properties like RG, something you could use to validate experimentally. Are we getting the right size? Maybe either for an aggregation of the protein emulsifier alone, or if it is an emulsion and you have an oil in there, can you get the RG right? Usually, at least for nano emulsions, we can do this reasonably well. And then you can start thinking about, adding more and more complex formulations. And so in this case, we're going to a coarse grain timescale, and we're moving up to much larger simulations. We're adding in a co-emulsifier, and, running these for even a microsecond, we start to see that this thing's assemble. We don't see the standard lecithin only behavior. We do see it almost forms a bicontinuous emulsion with just 2 components, which is really interesting. There's more work to do in this realm, especially with these simulations, run them for longer, change the various compositions. But what it shows us is that, just by changing simple things like solvent or adding a co-emulsifier, we can start to predict different types of assembly properties, which gives a very deep understanding of what's happening in your system. It may not be the same as running it as a reactor scale or at the manufacturing scale, but if you're trying to rationalize a behavior that you don't understand, either during pilot phase or production phase, or you're trying to make changes, these simulations can be used to either lower the costs by reducing the number of experiments you need to run, or in the case where some of the very high res techniques don't have the resolution to rationalize why you're seeing a certain behavior. Simulation usually adds that extra level of insight without having to, try a lot of really expensive experiments or make, new formulations that may or may not be easy at the end of the day. And so that's all I have. I'm happy to take questions. This is a newer project. It's evolving. We've done this with tons of other surfactants. We have a lot of other applications. More than happy to talk offline about. But this is just interesting because it's moving towards as as the food and beverage industry and cosmetic industry especially want more and more natural ingredients, and people are looking towards alternative sources for emulsifiers, these protein applications are becoming more and more interesting. And so we're going to see more and more of it as time goes. And so having a very good understanding of how they behave at a very small molecular scale can help really drive decisions or new product development at a much larger scale for everyone else. And so that's it.
All right, and we're back. Jeffrey, thanks for that. Definitely a different look at things. Could you share a little bit, and you touched on it right at the end as you were winding down, sort of where have we been 5 or 10 years ago, that kind of took us on the road here, and then maybe in another 5 to 10 years, where will your market be?
It's right. So good question. So 10 years ago, the way either the theory or the computational power, those simulations, the idea of even running a microsecond, which for a lot of people on this call, I'm sure seems insanely short. That was just not feasible. You ran a 20-second nano simulation. You really had to have your system assembled the right way. So if you had an emulsion, it had to be as a micelle, or you were never going to simulate that assembly. So, now that we can start to touch on these things, I think that, 10 years from now, following closely to Moore's law, you'd expect that we should be able to simulate emulsion foams or get very close to that. And the physics should be even better. So if there are small tweaks that we have to make now to a specific emulsifier or a specific emulsion to get the right experimental properties to match, we'll probably have much better force fields too down the road, where it'll just work for almost any realistic chemistry that one might want to use. So bigger and longer, I guess, would be the easy way to answer that.
Great. Question from the group: what is the molecular weight limit of a protein for the simulations?
So there's no molecular weight limit. Usually you can predict anything. So there are some proteins where they don't exist as a monomer. They exist either as a fibril or some, amorphous aggregation. There's no real limit. It's just when you get above maybe 20 nanometers, which is usually even dystrophin, I think you can fit in there, which is one of the largest proteins in the genome. You could probably do all of them. It just depends on whether you need to add 10 or 20 of them to get a realistic assembly for the property you're interested in.
Okay. All right. That's all the questions we have. Jeffrey will be back later for our roundtable Q&A. Thanks, Jeffrey.